Hi, everybody. It's Dr. KSS. It's a little after 1 o'clock, Wednesday, March the 3rd. It's a really special presentation today. We have a venerated guest. We always have venerated guests, but an unusually venerated one. And I think this is going to be a very lively discussion. And the audience is bursting at the seams already. Remember the poem, Yesterday Upon the Stair, I saw a man who was not there. I saw him there again today. How I wish the heck he would go away. Our guest today is the Edmund Safra Chair of Neuropsychopharmacology at Imperial College London, Dr. David Nutt. He's a consultant to working with our old friends at Algernon in their pursuit of DMT, dimethyltryptamine, or Dimitri as we called it earlier, as a post-stroke recovery agent based on some interesting and I would say sound lead-in data. And today's guest, along with Chris Moreau, is genuinely a world expert on this space, on psychedelics, psychiatry in general, psychotropes in general, and he has a storied history. I don't want to steal any of his thunder because reading his biography really is a bit of a page turner, but he is not a man who is shy about butting heads with authority when data and policy conflict, and when empiricism and good sense don't seem to match up very, or perceived wisdom don't seem to match up very well. Mm-hmm. And I'd like this to be really a fun, memorable session. I want to say at the outset, Dr. Nutt, I hope it won't be the last time we'll see you, but I want to make the most of the session and really have some lively chatter. Sure. And I'm sure our people will be forthcoming with the questions. In fact, I think they're already pumping them in here. But
take it away.
I wanted to lead out with a scenario, true story. About 2 years ago, pre-COVID, and it's something I've always wanted to ask of somebody of your stature. I have an uncle who was a man of very superior intellect, late 80s. He was a guy of multiple national high security clearances because he did avionics for their air forces. Mm-hmm. He was a top level Air Force guy, and he's getting on in years, and called me one night and said that I've not been feeling very well for several days. I seem to have something like an upper respiratory infection, but I'm seeing things. There's two guys over there across the room. I don't know who they are. Mm-hmm. And I said, well, do you feel menaced by them in any way? And no, they're just there puttering, working. And I said, well, I suspect, you need to be seen tonight. I suspect you're hyponatremic and you've got some brain swelling because you're ill. Mm-hmm. And then maybe one nerve is abutting on another and kind of, arcing out and bleeding signal into a domain where it shouldn't be. But, for as long as I've been in medicine, I've wondered about hallucinations and, what you can teach us about those. I mean, are they stored memories that are being bled off, retrieved and sort of reprocessed? Are they de novo? Is the dreamlike experience, can it in a proper way be called hallucinatory? Why is it that agonism of a receptor like 5-HT2A, overagonism would lead to hallucinatory activity? And other things, I kind of want to tug at your shirt sleeve and answer all these questions, please, because they've been piling up for years.
How long have we got? I've only budgeted an hour. I mean,
sorry. No, I thought you were going to stay the week with us, Dr. Nutt.
Yeah,
sorry.
Okay. Well, I mean, they're all wonderful questions. So, a very interesting. Just start with the man, the 80-year-old man. One of the interesting phenomena we see as people get older is that they do hallucinate more, and it often happens as they're going to sleep. They often get more vivid, particularly images of people they've never known. But and that eventually it becomes a syndrome. It's called Bonnet's syndrome, which is actually can be quite challenging because often these people then get in the way of their lives rather than your friend was just observing them in a rather sort of detached way. But begs a really fascinating question, and the answer I think is going to lie in understanding how psychedelics work. So we now know from our imaging studies that psychedelic drugs working on the 2A receptor, they disrupt a particular group of cells, which are called the layer 5 pyramidal cells in the cortex and comparable cells elsewhere in the subcortical regions. And those are the neurons which pull together all the crosstalk in the brain, because as you probably know, the brain is a kind of two-dimensional computer. It does the cortex that have a thing called cortical columns. So almost all your primary processing is done in these cortical columns. But then to make sense of everything, to kind of integrate the fact that I'm seeing you and hearing you, you've got to bring the two bits of the brain together. And that's done by these layer 5 pyramidal cells. And it turns out that those cells are the ones that are loaded with the 2A receptors. And when you give a psychedelic, you basically disrupt their functioning so that the brain works differently. And the reason you get hallucinations under psychedelics is it's a bit more complicated, but vision isn't a photograph. What you see is a reconstruction by your brain of what of the photons that have gone into your eye and your retina, incorporated into some kind of schema that your brain has already developed as to what's there. So I'm looking at you now. My brain has already worked out that you're you. And if you were to turn into a tiger, I probably wouldn't even notice, because why would I? I mean,
right.
I know who you are. You can't turn into a tiger. Therefore, I won't even assume you could. So the brain is what's called, it's a predicting organ. And these predictions are driven by these layer 5 pyramidal cells. And if you disrupt them, then you get different predictions, i.e. you see things which aren't there. You see hallucinations. So that's the first thing. The second thing to say is, of course, when people get into delirium, they often hallucinate a lot. So what you were thinking he might have a low sodium delirium, but there are many other kinds of delirium. And one possibility is that infective delirium might be due to the body making, or the microbiome in the gut making, these 5-HT2A like drugs like DMT, which actually get into the brain. And
really?
Yeah,
wow.
and in fact, we're now using blockers of the 2A system, particularly Risperidone. They're quite effective at treating delirium. So it may be that these receptors we're looking at all the way through are kind of central to both the subjective experiences of dreaming, of delirium, but also the mechanistic changes we see in terms of giving people psychedelics. And that's really important. And I mean, when Chris first approached me, I think it wasn't Chris, one of his team said, would we want to do DMT in stroke? And I said, well, that's pretty crazy, because stroke people probably don't want to be having psychedelic experiences. And he said, no, we don't want them. We want to give a low dose. We want to tickle up these neurons and make them regrow. We don't want to turn them into give people a flurried hallucinatory state.
Why is it, or is there a simple answer to the fact that auditory hallucinations are commonly the province of schizophrenia, but visual hallucinations, yet again, something different, different set of processes, delirium and whatnot. Why should they be so compartmentalized? Or is it compartmentalization?
That is a great question, and really interesting. So I think the core question is why schizophrenia is so associated with auditory hallucinations. And we know the mechanism. We know the mechanism of auditory hallucinations. We know it's because in schizophrenia, so maybe I've talked about vision. Let's talk about hearing and speech. When you are planning to say something, the part of your brain which is making you say, inventing pulling together the words,
right?
As you start to speak, it suppresses your hearing center.
Huh?
And that's so you don't get confused by, you don't listen to what you're saying. Otherwise, you kind of get. In schizophrenia, that suppression doesn't happen so well. But we don't know why it doesn't happen so well. I don't think we can attribute it to the 2A receptor. It must be some other system that's abnormal, and in schizophrenia that is leading to this. What we actually think now in schizophrenia is that those voices are in the same way as I was talking about the syndrome. Your friend was seeing things that aren't there. People. The voices in schizophrenia are the auditory cortex hearing things that are kind of created in the brain, but aren't really hearing. Then the really interesting question is why they are so hostile and negative. And to explain that, you've got to go back to kind of psychodynamic processes. They're self-critical thoughts, and some of them come from parents abusing them. Some come from guilt and sort of depressive in nature. So you begin to get much more into a psychotherapeutic interpretation of the content rather than a purely biological one.
Those who've never had the experience of walking in on a relatively unknown patient and sort of picking up on the fact that they are attending to auditory hallucinations and sort of winning their confidence and actually getting them to sort of tell you outright what they're hearing. And you're right, it's sort of it's bleak. It's horrifying in some cases. It's really yeah.
Often leads to suicide as well. I mean, the voices can be so destructive. And you can imagine having a running commentary in your head all the time about how shit you are and how useless you are and what a wanker you are. I mean, people do kill themselves because the voices just drive them mad.
Yeah, just to silence them. Is that the 5-HT2A antagonism? Is that the only mechanism by which risperidone is doing? Say, when you use it as an augmentation therapy for refractory depression, is that the only thing it's really doing? I didn't know it acted that way.
Yeah, I believe so. This is my particular theory, but risperidone has twice the affinity for the 2A receptor than for the dopamine receptor. And of course, it was invented to be a dopamine receptor antagonist to treat schizophrenia. Right. But they also put in the 2A antagonism thinking that would minimize some of the extrapyramidal side effects, which it does a little bit, but not much. But I take advantage of the 2A antagonism, and I use it in low doses to treat delirium, and I also in low doses to treat aggression. And that, I think, is why it's got a license in many countries to treat aggression in autism. And I think that's also a low dose effect with the 2A receptor.
Wow, fascinating. I mean, I gather, for not, sort of fleshing out a number of topics here, but I to return to the company hand Algernon and this program, what are you aboard this train? And do you think this is a worthy pursuit and an auspicious pursuit, what they're undertaking?
Well, they're not paying me enough to be kind of corrupting my soul. So yes, I do. That's right. So absolutely. I think as I said in previous talks, I'm envious that they came up with the idea. It's a really clever idea because, we've been working on how to look at synaptic plasticity, brain plasticity. We've got great theories for 10 years on how these psychedelic drugs change your mood, make you undepressed, and how that persists. But I never thought of using it in stroke. It's a very clever idea.
Yeah.
And I don't think we can fully take credit, and won't fully take credit. I mean, there were the two components were the sub-psychedelic dose. Mm-hmm. That was established as having neuroplasticity and neurogenesis by Olson in his in vitro work. Then he did an in vivo study showing the sub-psychedelic dose was actually active in reducing depression in that animal model. And then the Nagy group from Hungary that did the ischemic stroke model, where they tied off part of the blood flow to the brain. And they pre-treated, I think, a few hours or a day, which is kind of interesting. That would be immensely challenging to do clinically. But at 30 days, you had this reduction of dead cells, this infarct volume, and also a reduction of motor function, almost 100%. So we're Edie Fresca had been talking. These are people very passionate, like Dr. Nutt, about the potential for psychedelics and also in this case DMT. So Mark Williams and team pulled it together, but we're not going to take credit for all that early research, but we want to drive it forward.
Chris, when DMT does exert an effect in animal models on depression, that's very curious. I mean, how rapidly does that exert itself? How do we see the animal in question become more animated? And are we talking days, weeks? How quickly is it?
It would be a great question if we had our other science member here to tell you exactly about pulling that study apart. But there is this, it's either a rat or a mouse model for depression. They can depress the animal and look at its function, and then introducing DMT. I would suspect because DMT has a half-life of 15 minutes, that I think they were looking really at a bolus dose, but Dr. Nutt, can you comment on that?
Well, I think let's say a few things about psychedelics and depression. So the animal literature is slightly, animals and depression are kind of slightly complicated things because we can't really ask them how they're feeling. So the motor data for stroke is really compelling, because it's obvious if a rat has had a stroke, you can see it's not moving its paw very well. But so in depression, actually there's certainly, it's not a uniform finding that psychedelics improve depression in animal models. All right. But in humans, it's a very powerful finding. And the reason is that it disrupts depressive thinking. And so let me just be clear about this. So what Algernon are exploring, they're not exploring that phenomenon. They're not interested in the disruption of neural function to lead to a different kind of insight, which is what a psychedelic insight is. They're interested in the other aspect of the 2A receptor, which is the neuroplasticity. Now, neuroplasticity has long been implicated in the effect of different antidepressants. And it comes on slower. It's not something that comes on instant, but within 24 hours, you can see in a rat neuroplastic changes, more synaptic spines, bit of bigger dendrites, etc., from ketamine. And there's now evidence the same can happen with psychedelic drugs, the 2A agonists. So what we are thinking, and this is why these particular drugs are really intriguing. Not only do they, in depression, they don't only reset your thinking, but they give your brain the ability to encode and register and stabilize that new thinking by these synaptic plasticity processes. And those will be the same processes as we would hopefully see in humans who've had a stroke. You basically make the bits of the brain which are recovering, desperately fighting off the toxins and the dead bit, allowing them to grow and become more functional so that you can stabilize or improve and then stabilize your motor function.
So follow-on dosing doesn't destabilize the work that you've just accomplished then, right? Or one hopes.
Now that is a great question. No one's ever done those experiments, but I can tell you, that we have just finished a two dosing, 6 weeks, 2 doses of psilocybin, 1 at times 0, 1 at times 3. Look at 6 weeks. And we've done that in a comparison with an SSRI, escitalopram. And what you see is that most of the benefit of the psilocybin comes in the first dose.
Interesting.
It doesn't go away after the second dose. So yes, I can answer your question categorically. The second dose doesn't disrupt the effect of the first dose. Right. But there's not a great deal of added effect on the second dose. Now that could be a floor effect, or it could be that you've already stabilized these new thinking processes, and so that you can't improve on them.
That's it. For therapeutic administration of an agent like that, in your country, would you seek to confine and supervise the patient for that interval, or can they go home? Or
well, we use psilocybin as our main psychedelic for this reason, which is that when you take orally, the trips last between about 3 and 4 hours. So if you start in the morning, people can go home in the evening. So that's the first thing. But all our administrations I've done in hospital. For safety reasons, not that it's unsafe, but just because it's so early in terms of what you might call the modern science, that we just don't want to run any risks. But also, that's where we work. So they come to us, to our hospital. Our therapists are there. We always have at least one therapist. Usually we have 2 therapists present during the experience, not to do therapy, but just so that people are reassured that if anything goes wrong, they're there to be, and they're being nurtured and looked after. The work, the therapeutic work, is done the next day in what's called an integration session, when people because people can't really communicate when they're in a deep trip, because things are happening to their brain which are so unusual. They're kind of engrossed in those. But the next day they can talk about them, and then they can really begin to make sense of, for instance, why they were depressed in the first place, and look at the thinking that maybe the maladaptive thinking they've put into thinking about something that went wrong, and you can say, well, maybe it wasn't that bad. Maybe actually, you did get, beaten up a little bit by your brother, but you're still alive and you still love him. So kind of break free from that and see the positives.
Yeah, shaking it off like so much dried mud off your boots and moving on.
Yeah, absolutely. But yes, go on.
In our case, our MTD, our maximum tolerated dose, will be trying to be just sub-psychedelic. So we're not expecting to have to keep people overnight, and the protocol is just being developed. And but we sort of know, I think the maximum tolerated, the maximum dose established by Dr. Strassman was in the 0.6 milligrams per kilogram, where people were really tripping out and they couldn't even recall their memory of the psychedelic experience. They couldn't really communicate it. And I'm just getting my way through the spirit molecule. So this is the early part of the book. And then sort of the sub-psychedelic is in the 0.2, 0.3 milligrams per kilogram. But David, do you want to talk a bit about how you were thinking we'll use PET scan during the phase 1?
Yeah, we can. So I think it's important to, yeah, I mean, but there's what we've got to qualify this, Chris. We've got to say, we're going to do this very carefully, because what we know is a sub-psychedelic dose in a normal healthy volunteer may not be sub-psychedelic in someone who's had a stroke. I mean, stroke patients often have a degree of, we're going to do it. We're not going to, well, we're debating at present. We don't even know when the best time to do it is, do we? We don't know whether it's to do it when they're having the stroke or, up to a week or so after. So that's still a question to be resolved.
Yes.
But so we're going to be doing a very careful dose finding studies initially in healthy volunteers to make sure we know what a sub-psychedelic dose is, because this is an infusion. So typically, Strassman just gave a shot, and that gives you higher peak levels. So this is going to be a gentle infusion over maybe several hours. We haven't finally decided how long. And then we're going to, so how are we going to monitor the effects? Well, obviously we're going to ask people what they're feeling, but the other thing we can do is do EEG. And there's a very characteristic change in EEG cohesion as a result of these drugs. It's really very clear. It becomes the brain becomes very unsynchronized. The normal patterns begin to break down. You get what we call an entropic state. And so that's a measure, again, of an effect. So we can potentially, in the real world, we might even be able to have real-time measurements of the EEG and titrate up to a point where they begin to scramble and then stop, or cut the dose down. So using the EEG, as you might say, use blood pressure as a measure of an effect on the cardiac system.
Dr. For readers, thinking about psychedelics, that space of drugs in general, could you articulate kind of the case for choosing DMT as opposed to other potentially available agents for the stroke indication?
Well, yeah, I mean, the main reason for choosing DMT is it's as soon as you stop infusing it, it starts having an effect. So if anyone has a bad response, if you infuse LSD, which we've done, given an IV, then we've got to wait 12 hours till it's cleared, which is a long time if someone's having a bad trip. Whereas DMT, the effect just disappears. So I think that's a major safety benefit of it. And the same psilocybin, that's not so, but psilocybin effect wears off IV after about half an hour, but DMT is in this order of seconds, really, the effect is.
I remember in high school sort of being exposed to the concept, not the drugs themselves, but the concept of agents that acted this way. And I remember reading that in the LSD trip that so many people experience it would come back from that experience having said that an entity called the Watcher was present throughout the trip. And it's been many years since I've read descriptions of LSD trips where the Watcher is alluded to, but there are some patients who I think have described things in those trips that occasionally were more terrifying than anything I've ever experienced while awake, and yet they ascribe their survival to the presence of the Watcher. Is there an anatomic or neurophysiologic correlate of that event? Is it the soul popping into the window there? I mean, is it
great? So why did Algernon come to me? You better ask Chris. But my supposition is because we have done the most detailed analysis of IV DMT, a lot more than Strassman's ever done, and we've done it with EEG, but we've also done it with fMRI. So we now have brain images of people who are experiencing what we call entities. They've gone from being a watchman to entities. And we've got people going into DMT trips, talking to entities being nice to them, helping them. We know when they're going in, we say, press the button when you meet the entity. So we're in the process, it's quite complicated data to analyze, but we're in the process of analyzing the MRI data to see if we can see what the brain signature of these entities is. So that's
But
the first thing to say.
the response to the projection of the entity is invariably a benign one, I think, is it? And I think some trippers, if we can call them that, wonder if it is a projection of themselves. Is that fair to say? Is it self?
Well, this is the real debate. There are plenty. Why is DMT such a popular drug? Because it much more reliably flips people into another universe. They go through the wormhole. They go into a universe which is better, brighter, more meaningful, more real. It's where they want to be. And that's why people keep doing it, because they want to be in this, basically it's a more heavenly kind of place. And in that environment, they some of them consistently meet their entities who guide them, nurture them, tell them, reward. It's a very positive experience, but not always. Sometimes they meet rather hostile entities. So my simple interpretation, most people believe that they are going somewhere else, to another dimension, another universe. We can't follow them there, but all we can do is see that they're still in the scanner. So my current interpretation is, again, a rather sort of Freudian one, that what you're seeing are kind of internal constructs of yourself, or friends or other people, or constructs like niceness or support. The construct is turning into an entity. But certainly when we're doing the psychedelic work in depression, people often have very unpleasant experiences. It's not fun, because they're working through the traumas that led to their depression. So it's we can't generalize about that. But accept that most DMT entities at least start off being pleasant, but sometimes if you keep going back to them, they seem to get a bit fed up and become a bit negative.
Does the entity map to a locus on fMRI?
Well, we're going to find out, and when we do that, half the world's going to love me and half the world's going to hate me, whichever I find.
We'll love you, I'm sure. I mean that, but
well, it depends entirely on your perspective. Are they in the brain or are they out there? There are, people believe genuinely, and I certainly can't prove them wrong, that they actually, their mind leaves their brain and wanders around the universe and meets the entities and comes back. It's very
that's not inconsistent with quantum mechanics as we now know it. So, is it?
Well, I'm not expert enough to comment on that, but it's been said that is perfectly possible. Yes.
Well, say more about the trial design and where you think this is taking us, and because really, one of the we're a site that talks about investing, and a thing that gives Algernon in particular staying power as an investment now. Not that it lacked that before; it had plenty of it, in spades. But this story has staying power, I think, because we're learning about ourselves now. Some questions that really haven't been asked before.
Yeah, but hang on, I'm going to chip in. I'm going to ask Chris to take over, because I think, just to emphasize, we are not going to give people who have had strokes trips. We do not want to do that. It might be that it's not good for them. So that's the first thing to say. So we're looking at the neuroplasticity of these receptors rather than the immediate tripping effect. That's the first thing to say. The second thing is, and I'm not sure whether we can, I don't know how much is disclosed yet, because I don't think the actual protocol, Chris, is fully in the public domain. So I'm going to leave it up to you if you want to talk more about what the design is, because I think it's still quite experimental.
It's still in process, but at a high level. And in fact, we're just planning with Hammersmith the IV study to make sure that DMT doesn't stick to the inside of IV tubes. To some extent, because it's not a bolus injection, this is a continuous flow. There's that very early part of it. So then there's the phase 1 study, which is to determine, should we be dosing it for 1 hour, 3 hours, 6 hours, 12 hours, holding them up at that dose. As David said, the good thing about DMT is that it has a short half-life and you can turn that pump off. On a bigger picture, we haven't seen hemorrhagic in vivo data yet, just ischemic. And there is some evidence that DMT raises blood pressure. We talked a bit about this on the first show. And typically right now, if your partner finds you and you're not looking well and you're speaking in a confused state, and they go through the process, hold your hands above your head and ask some questions. They call 911. They really can't treat you unless they have a CT scan to know if you have a ischemic, which is a blockage, or a hemorrhagic, which is a bleed. Because the 85% of people have an ischemic stroke, the treatment is a blood thinners, clot buster. That would be very detrimental if you had a bleed. So only about 10% of people are getting that CT scan within that 3-hour window. It's getting better. It's not just trauma centers, but it's actually, if you're with somebody when you're experiencing symptoms, could happen at night when you're sleeping. How do you get somebody to the hospital in that treatment window? Because for a TPA, you have to be within 3 hours. And then only about 10% of people respond. After 24 hours, it's prayer. There's not much there for stroke. And I just got off the phone with an analyst this morning who said to me that he personally felt this was a graveyard of failed attempts to try to treat stroke. And he called it high risk. And then we talked a bit, and I said, that's, to me, there's opportunity.
The bar is set so low, Chris. I mean, anything you nudge above where the bar is, that's success. That's easy. I don't want to say it's easy, but you see where I'm going with that notion.
And we can get into our plan is to be into a phase 1 trial in less than $3 million US. So I had talked to him about the risk of a new chemical entity, bringing that through from discovery through it to a phase 1. Only 10% of drugs make it, and you're talking 20 to 30 million. Never mind the cost of phase 1 and the risk. So we I'm not sure who won that argument, but so in terms of treatment, what is the best time? As David said, we don't really know a lot. Should it be as quick after the insult as possible? Will it be negative to hemorrhagic patients, in which case we have to wait? We'll be looking at blood pressure during our phase 1. Can we mediate maybe a channel blocker to reduce it? We have to look at that, and I'll turn that over to the experts. For our phase 2 trial, the less complicated path is the rehabilitation path.
Yeah, absolutely.
Because there are complexities to the phase 2 in terms of treatment arms. First 3 hours, post 3 hours, TPA, hemorrhagic, and time period. Whereas post-stroke, after the insult, 24 to 48 hours, they start rehabilitation. 85% of people have some deficit. So that would be the first and quickest. We'll do them parallel, but if we can infuse DMT to patients who are trying to recover, and specifically CIMT, which is constraint-induced movement therapy, they literally tie off the limb to encourage using the limb that's been affected. And we have an announcement, won't be made today, but shortly, of a global expert in CIMT that's now joined Algernon's team, and to help us with our phase 2 trial design. And so that, and basically that could be a 30-day trial. So that would be very quick. And then while we're working on the other phase 2 for acute treatment. So that's the big picture of where we'd like to take DMT.
Chris, just to, and Dr. Nutt, to attend to a couple of comments and questions coming in. Chris, one reader, Athi, is asking, in effect, if there were a single point of origin within Algernon for a decision to go in this direction, what was it? If you can comment on that, if it can be traced back to exactly where it began.
It happened in early December. As I've said on many interviews, Algernon is not a one-trick pony, and we've talked often about the fact that 50% of phase 2 trials succeed. And so most companies like Algernon, if they've started with a new chemical entity, have spent 20 to 30 million, and they're lucky if they have one compound in a phase 2. We're looking at 2 to 3 million to take a repurposed drug into a phase 2. It only makes sense to have a couple of drugs in phase 2 trials.
Sure.
That way we're increasing everyone's chances that we'll have a hit or a win. So in early December, I'm connected to the capital markets. I know that psychedelic drug companies have crazy valuations. And I sat down with Mark Williams and I said, Mark, let's look at psychedelics. And he said, not a chance, no way. We're not doing the mushroom thing. And I said, easy. I said, let's take a pharma approach And over the Christmas period, he really dug in on psilocybin, psilocin, DMT. He looked at a number of different options, and he was fascinated with the stroke information. He had a background in stroke. His previous firm, Diabetica, is in a phase 2 trial. So we had all this info on stroke. All of this preclinical data was coming out, and he quickly identified key opinion leaders, including David Nutt. And, Mark, and there was no ceremony. David took an email and said, I'll be glad to talk to you. And that's what happens, I think, often. He probably gets lots of emails, and he'll give you the benefit of the doubt for 5 minutes, and then he'll either agree that it's a good idea or it's not, and he'll move on. And then the biggest decision was, could we get an intellectual property position? Because all the companies in this space, you've got naturally occurring compounds. You can't file composition of matter. If you make a change to the structure that's unique, you're back in preclinical. You have all your tox work and genotox, and you might end up on the heap of failed drugs. So we were able to make a slight change to DMT. That's why we're calling it AP188, which is its molecular weight. That is allowed us to file IP for a new form of DMT. We don't believe it will cause us to have to go back and start at the beginning. We filed dosing patents, formulation, and combination therapy. So it happened around late December during the Christmas break. We had some confirmation mid-January on intellectual property. We reached out to key opinion leaders like David and Rick and Dennis Choi, our stroke expert, and we announced on Feb 1. So it moved quick, but it was very organic in its process.
Someone is asking about the use of LSD with some concurrent medical circumstances. I think we need to take a pass on that question because that patient is clearly more medically complicated than face value. I would proceed with caution. Chris, someone's asking about, quote, anything about the 5th. I'm not sure. Martin, you're asking that. I don't know what you mean by the 5th.
Yeah, March the 5th, we put a news release out that the database would be locked for our COVID iPhone trial. And no change to that. That's when all the queries are done, the data is locked, and then the internal review from Novatech and the statistical review group starts. So I'll come back to the market a short time after and update on when we're going to have the final data in Algernon's hands, and then when we can disclose it to the public. And just for clarity, Doc.
Sure. Yeah,
on issues like Mark Williams, which I'm open to discuss, also our current financings. At head office, we have absolutely no information on COVID, on our COVID trial. It's it's a bit of a, on the one hand, I want to answer all questions, but we don't have any access to data. If we had that data, it would be material within reason. We'd have a few days, we'd have to announce it.
And folks, remember, trying to just sort of help take care of a company we like. That's why I did the videotape that's up now, just sort of trying to preemptively answer a lot of these questions. If those questions are still coming to mind, please go watch that video if you would, because I think they're all answered there. But no, there won't be COVID phase 2B data today. And I'd really like to use Professor Nutt's time while he's here, because if he needs to break away for something else, we can field other questions later. But let's return to the psychedelic discussion and his expertise there. Dr. Nutt, how do you intuit this playing itself out? I mean, I hate to ask a question like, do you believe it's going to work? What do you anticipate? Because belief is not scientific, but supposing there is efficacy, where does Algernon go next with this project?
Okay, if we show it accelerates rehabilitation from stroke, I'd go global, but I'd probably start with America, US. Okay.
Will there be room for modifications of dosing or duration of dosing, or?
Bound to be. I mean, if we get a positive readout from the phase 2, there's going to be a lot of questions, but hopefully there'll be a lot of money to answer those questions, because that, you know, as you pointed out, and as Chris has mentioned, it's been a graveyard. I mean, it's funny to say, I mean, I did some stroke work 30 years ago with some barbiturate compounds. You know, you can make animals respond. It's really, really difficult to get positive readouts in humans. And the clotbuster drugs, they have a bit of an effect. I actually did a bit of work, or at least a bit of advising, around the old vampire bat venom, which is also another clotbuster. But doing the experiments in acute stroke is just too difficult, too expensive, too challenging. But this idea of accelerating rehabilitation, I think, is really, really very exciting. That's why I'm helping Algernon. And I can see beyond that. I get quite a lot of emails from people saying, you know, I've cured my dyslexia with psychedelics. And I'm thinking, it's interesting, isn't it? You know, I mean, you know, maybe there are other neurological syndromes which you know, we might be able to, you know, use DMT on after we've shown it does help, at least in stroke.
Again, it's not exactly a fair question to ask, but I'm going to fire this one at you because you know the space better than anybody that we can speak to live. You know, let's assume it does work. You know, that there is efficacy seen. Is it fair to ask you to comment what kind of effect size you think we might be looking at? I know it's not fair, but you know, anybody knows.
A significant one.
Okay, a meaningful one. Fair enough.
I mean, a clinically significant one. That, you know, that's the crunch, isn't it?
Yeah.
You know, we don't. You know, there's not much point in showing, you know, that somehow in some imaging measure, but people basically need to get their limbs or their tongue or whatever moving better than you would predict from normal recovery. And then, of course, it's going to have to be a controlled trial. I mean, you know, it's going to have to be some kind of placebo head to head.
Chris, do you think that hemorrhagic stroke is permanently verboten for this drug, this indication? Because really, the two processes do have a way of coalescing at the end, right?
I think it's just strictly the only thing that we've seen, and David could maybe talk to other issues about how the liver metabolizes DMT and contradictions with other medications. But the only thing that we're concerned about is this slight raise of blood pressure when you take DMT. And in fact, I haven't had a chance to dig in, and I haven't talked to David about it, but we were talking in our previous webcast that I think post subpsychedelic, you've got an increase in blood pressure because of the experiential part of the treatment.
Yeah, exactly.
But in the subpsychedelic, there still may be a receptor that's sort of increasing pulse, but we can monitor that very closely. We'll find out. We either decide if we can use DMT along with the channel blocker to reduce the blood pressure. If we find out that it's not that critical, and I sent a paper to David from a week ago in Australia where a group showed that the maximal time for neuroplasticity, neurogenesis, is about 14 days after the insult. If we don't have to worry too much about that 3-hour window pre-CT scan, then even the study would be a little easier, because we just say right off the bat, we're going to start with ischemic. We're not worrying about hemorrhagic. Right. But we don't really know. There's lots. The good thing though is that we're going to be able to get this very quickly into a phase 2 trial, so we can get these questions answered. And that's the Algernon model, and that's one that we're just about to conclude our IB for DMT. There's a tremendous amount of information about the drug. We don't believe we have to be concerned with doing talks, because there are a number of phase 1 studies, even back in the 90s with Strassman, that went on without that. There's lots in the literature about the safety of the drug. So we think about 5 months for Dalton to manufacture cGMP material for us. Meanwhile, if we get it sooner, and I'm always pushing, then we'll be able to start that phase 1 sooner. Once we have the phase 1 data, it will help finalize the protocol for phase 2, or our multiple phase 2 trials, rehabilitation, acute therapy and off to the races. So that's, David, you want to chat a bit about maybe your concern about DMT with other drugs?
No, I'm pretty relaxed about this because we're losing a load of. I actually be surprised if there's really any significant effect on blood pressure or heart rate. I'd be surprised. I think your question about ischemic stroke is fascinating because, of course, in the end, the brain damage from ischemic stroke and from hemorrhagic stroke is the same. It's basically cells get killed, and then the dead cells spew out lots of metabolites, which then kill the other cells. So the process, of nurturing the non-dead cells is what we're about. And I don't think there's any likelihood that DMT would actually cause a clot to sort of rise and to suddenly exacerbate a hemorrhagic stroke. So I'm relatively relaxed about that. I mean, during the acute bleed, it wouldn't be good potentially, but afterwards, a day or two or a week after, and as Chris said, there may well be a period where a responsive period up to 14 days after a stroke when you can get some kind of rehabilitation. The paper was alluding to was a fascinating one. It was where they were using electrical impulses to the brain to facilitate it. And my response to him was that's interesting. It would probably be even better if they gave the electrical impulses on top of a DMT infusion. And that is an experiment we can potentially do in non-clinical models.
Yep.
I want to say one other thing. Chris mentioned it, and I didn't have time to talk about it previously. We do now have a PET. You know about PET imaging, positron emission tomography.
Yeah.
We now have a PET tracer that we don't know, but we're using it to see if we can actually measure synaptic plasticity. Measure.
Oh gosh.
And we're in the process of scanning after ketamine. We know ketamine causes synaptic spinal growth. We've got that protocol up and running. We're starting scanning now at Imperial, and when Chris gets enough investment, then we really want to scan these stroke patients as well and see if we can see regrowth in their brains.
Dr. Nutt, that leads right into another question. I mean, could the competitive landscape for this project at some point include someone who wants to try ketamine or esketamine as a post-stroke recovery agent, given the ostensible kind of neurotrophic activity, if we can call it that?
Yeah. Okay. Well, that's a really clever idea, and I don't know that Chris can actually stop the pattern. I don't exactly. It makes the concept of rehabilitation through products which release brain-derived neurotrophic factors obviously would extend to ketamine too. It's an interesting concept. I mean, the fact that ketamine's been around for 50 years and no one's noticed it argues perhaps there's nothing there, but it's never been, to my knowledge, not been formally tested.
I mean, there's a certain dysautonomia that can go with dosing, especially I think in older people. But I would assume that as we get better in our art and more accustomed to using that routinely for refractory depression, we might get better in our groove at how we manage those patients and could use
one really important. This is a really important point you raise. Ketamine is a psychedelic. DMT is a psychedelic. The difference is, and ketamine treats depression immediately, and we presume DMT would. It's not been studied, but certainly psilocybin does.
Right.
But there's a fundamental difference between ketamine and psilocybin. The ketamine effect lasts at the most 2 days.
Okay.
The psilocybin effect can last for 2 months. And that is part of the reason I think that the 2A receptor is involved in reprogramming the brain. Whereas what ketamine does, ketamine is just like, it's like a snow globe. You shake up the brain and the depressive thoughts disappear.
But
they've not gone, they come back. Whereas psilocybin disrupts them and then allows you to put down non-depressive thinking on top of them.
Okay. I did not know that, and so it does make you wonder whether these patients queuing up to have their ketamine, treatments at $400 a pop in the United States they're certainly not getting their money's worth. The ketamine clinic in the countryside has become sort of a fixture, staple of life in certain areas of the U.S.
Ketamine is a very good business. It's a very good business model for doctors.
Uh-huh.
But I think psychedelics will be considerably better for patients.
Yeah, agreed. Okay.
Right. Unless some company can get an intellectual property position, there are many drugs that could be repurposed. But the big challenge is a phase 3 trial and the cost of a phase 3 trial. And just speaking from experience, big pharma, they really need to have an IP position. And if it's going to be highly commoditized or it's widely genericized, there's just no interest. And it's I think maybe that's a discussion for another day, that some sort of a program should be developed for funding compounds that could be efficacious, that don't, that can't be billion-dollar drugs. But there's just so many drugs, I think, that we've come across just ourselves, where we looked at and said, wow, this could be very effective, but nobody is going to write the check because somebody else will just jump in and genericize it. It's an issue.
Folks, a little station identification. We're live with CEO Chris Moreau of Algernon and our guest, Professor Dr. David Nutt, who is Edmund Safra Professor of Neuropsychopharmacology at Imperial College London, a leading global expert on psychedelics, and he's taking our questions about that space and Algernon's intention of pursuing DMT as a therapeutic for stroke, for driving synaptic and neurotrophic recovery there. Are there, let's see, let me check my console here. We probably do have some other questions here. Hang on, scroll down.
I think you can add to Dr. Nutt's background, and one hell of a nice guy.
Karen Podolka is asking, I've seen patients continue to make progress for 6 to 12 months with therapy following stroke. I wonder if it would be useful to continue some extent of DMT infusion as well.
Well, this is why it's exciting, isn't it? Completely, utterly new concept in stroke. Rehabilitation, a pro-rehabilitation drug. I mean, how interesting is that? That's why I'm interested. If it works at all, then it's going to be extended and we're going to find out how to optimize it. Maybe you need a bolus every week as part of your therapy when you're having your physiotherapy. There's also, there are many, many potential models that we're going to have to explore, but we can't explore them until we've got funding, and we won't get funding until we got the preliminary data.
Karen, by the way, is a career RN nurse here. She's asking about specifics, if one could provide them, for what electrical impulse therapy would look like. How would you practically administer that in a setting like this? Because I don't know that we've seen, we haven't seen that in this country.
You do it through transcutaneous magnetic TMS.
Okay.
You just zap, you put the magnetic field in, the magnetic field depolarises neurons and neurons fire. That's actually, I've been asking my TMS colleagues to do TMS in someone who's got a psychedelic on board to see whether we can actually show it changes that sort of synaptic excitability.
Yeah.
But they're not so interested in pharmacology. They're physicists, really.
Dr. Nutt, are you good on time to be with us a bit longer, or do you need to be sprung loose too?
Got 5 minutes.
Okay. Folks, let's make haste and gather questions if we've got them for Professor Nutt. Chris, other comments from you?
Not specifically. I'm just actually I'm anxious to get our work started in terms of getting our phase 1 trial. We really tried very diligently to get access to clinical grade material. Charles River Laboratory is doing a number of pre-clinical studies for us to help us direct our phase 2 work. But they're accessing research grade DMT. But there are really only a handful of manufacturers globally that could take on the job. There's one in UK. Interestingly enough, because this is such a commoditized space, some of the early companies tried to tie up the manufacturers. It just made sense with contracts that wouldn't allow them to manufacture psilocybin or psilocin or DMT for someone else. That's a classic move, because you've got to try to tie up supply. And a couple of companies we did reach out to were under restrictions, and and they'd have to recreate, or maybe they've licensed out a certain part of the process. But I think DMT is a fairly, maybe 1 or 2 step synthesis process. It's not that complex, but it takes time. There's a process quality, there's stability that will be needed. I think we can take the API and probably turn it into a soluble right at the site in Hammersmith. They have that capacity to do a sterile screen. So we're going to move this along as quickly as we can, but not for this just the sake of expediency. And we'll be working closely with David on every aspect of our plans to make sure we're asking the right questions and that the study is the right study.
Chris, we, as always, really are grateful for the way you keep, so many of us are Algernon investors, the way you keep us in the loop here and are a regular guest. Dr. Nutt, I really am begging you, please, to come back again this year, because I'd like a longer session with you with some more questions. This is very, very edifying. And sorry,
I'd be very pleased to. Yeah.
That would be awesome. And Chris, we've got, I guess, a data read impending. I'm sure you don't want to commit to a time, but we'll be having you, I guess, back fairly soon when that verdict is in. And folks, there will not be a webcast this Friday. And in fact, we're going to take a couple weeks off to catch up administratively and on some writing, and then resume late month with a couple of very important webcasts. So we're behind and kind of need a break, like many of us do. But this has been a fabulous experience. I am really grateful, Chris, for you getting our guest today. Very generous of you. Very generous for you to spend your time with us, sir. And really look forward to seeing you back. Open ticket on our open space on our dance card, as we say here. And
I just want to let me just say one last thing. I have to say, I have been impressed by how fast Algernon worked. Having worked, I've done a lot of consulting for big pharma. I mean, they wouldn't have even put together a project outline, concept outline, and these guys have already got the trial up and running. I mean, they are pretty impressive, I have to say.
It's their leitmotif. And from day one, meeting Chris and Mark, before we ever began covering them, like, how do you not cover them when they deliver line by line on everything they say they'll do? They don't let the grass grow under their feet. We're proud to back them for that reason, because they don't lollygag and they deliver. So, and Chris, great on how you've handled the changing of the guard. I think we got that behind us, and it's all smooth, all good. And so,
thank you so much.
Thank you, David.
Thank you. We'll see you online in the BioPub. Thanks for joining us. Have a good afternoon, a good weekend. Thank you.